Our next speaker is Marcel Walser. After receiving his Master of Science in Molecular Biology, Marcel obtained his PhD from the Swiss Federal Institute of Technology in Zurich, during which he developed novel screening methods for biotechnological applications. Since joining Molecular Partners, Marcel leads several research activities to enable antibody mimetics, DARPin proteins, for various therapeutic programs. Today, Marcel will present the generation process and preclinical data of a trispecific clinical stage DARPin candidate directed against the SARS-CoV-2 spike protein. Marcel?
Hi, Peter. Thanks for the introduction. So, ready then to move on. Thanks a lot for the invite, and on behalf of Molecular Partners, I would like to present today the generation of anti-SARS-CoV-2 multi-domain DARPin molecules, which act as highly potent therapeutics in the COVID space. This is a brief disclaimer, and on the next slide, briefly showing our Molecular Partners team. We're a company with about 170 employees at the moment, and this picture was taken before COVID times, so everybody without masks. Here we see our pipeline. So besides the oncology franchise in blue and the ophthalmology franchise in orange, we're now focusing and discovering the infectious diseases, and in the COVID space, we have our clinical candidate, ensovibep, in partnership with Novartis to develop forward, currently in phase 2-3, and another program called MP423. Both are trispecific DARPin molecules which act and neutralize the spike protein interaction to the ACE2. I briefly want to introduce to the DARPin platform, as probably most of you are not very familiar with it, and Peter nicely introduced here that we're antibody mimetics, and the DARPin proteins come from ankyrin repeat proteins, which also occur in nature, and they are about 10-fold smaller binding proteins compared to monoclonal antibodies. The nice thing about DARPins is you can link them quite nicely with protein linkers, so genetically fuse them, make single chain molecules with multi-specificity. Usually they have very high affinities in the picomolar range and good target specificities. And the simple architecture and also the DARPins do not contain any cysteines, and this makes them very nice molecules to produce them in high yields in microbial expression systems. Usually we get around 20 grams per liter in a fermentation broth. For half-life, we can add some HSA binding DARPins to tune half-life, and usually we get up to 2 to 3 weeks half-life in human. Safety and immunogenicity is generally very low. A bit more to illustrate the binding mode of an antibody versus a DARPin. Here on the top, you see in orange the CDR loops of a monoclonal antibody binding to the target, relatively flexible loops. In the DARPin, that's a bit different. So the DARPin scaffold per se is very rigid. It's an alpha helical structure. And you see in orange the binding surface, and in the libraries, the orange residues would be mutated, and they're very well embedded in the scaffold, which gives a very rigid binding and also very precise binding to the target. This is quickly to outline that ankyrin repeat proteins are the nature's choice for multispecificity. There are several ankyrin repeat proteins which bind multiple targets by nature. And we use this principle to in the green field, solve clinical relevant problems. So using them for localized activities, for localized agonists. And also doing molecular handcuffing, so basically having two DARPins binding to different domains of a molecule and forcing it into a certain conformation where it's inactive. This was, for example, done with MP274, a molecule directed against HER2 to neutralize the HER2 receptor. And then also for escape, multivalency and multispecificity was used in Ensovibep, so our clinical candidate against SARS-CoV-2, to prevent escape for the virus. Quickly go into the DARPin generation process, how we usually generate DARPins. This more to also illustrate that our SARS-CoV-2 DARPins were not taken out of a patient, as the DARPins are engineered molecules. So here you see the DARPin generation process, and usually we start like depicted on the right side with ribosome display selections. Therefore, we have mRNA libraries with a diversity of 10 to the 12. So this very high diversity allows us then also to select very highly affine DARPin modules against the target. First, we select the mono domain building blocks, and then from that, after characterization of these monodomain building blocks, we move on into the screening for the multi-DARPin space. Here we can, within one reaction, generate multi-DARPins, which have multiple binding to different target domains quite easily. That's basically one single reaction for cloning 6 valencies into one molecule within one go. This can be a randomized step, so then basically screening out the best combinations, or a rational approach to assemble these molecules and these multi-specific DARPin candidates, which then, as seen on the right side, can solve a certain therapeutic problem. For that, we use this modular approach, having the different DARPin building blocks, either to assemble them into multi-specific molecules, but also we can play a lot with linker length, linker rigidity. This is especially important for T-cell engager platforms, but also for the SARS-CoV-2. This was very important to look at the linker length, that this basically the architecture of the molecule fits to the target molecule. Then, as said, DARPins do not have any cysteines incorporated, so that this allows us to attach a cysteine wherever we want within the molecule. So this can be in a linker sequence or at the N or C term of the DARPin, to then fuse or couple any toxins, or to make ADCs or make radiotherapy treatments. And all these properties then were used and applied to efficiently neutralize SARS-CoV-2, and this program was initiated about a year ago during the lockdown. And what we see here is the 3D picture of Ensovibep and how this is binding to the SARS-CoV-2 spike protein trimer. So the blue and reddish DARPins bind to the RBD domain, thereof the RBM loop. I come to this later. And in gray on top, you see the N-terminal HSA binders of Ensovibep. And this cooperative binding of the 3 DARPin modules binding to the trimeric spike protein gives very high potency, and we have also seen that it protects very well for viral escape. The half-life with these 2 HSA binders was found in human to be about 2 to 3 weeks, and we want to point out that we don't have an active FC mediating immune cell activation, and we believe that this can make a difference in patients which already have inflamed lungs. Ensovibep can also be produced microbially, and this manufacturing should guarantee large-scale production at decent costs. And also the small size and high solubility of the molecule should enable a bolus and sub-Q injection. This is the generation process of Enzovibep. It's very tiny. I zoom in soon. We used about 11 weeks from the initiation of the selections until in vivo proof of concept in a hamster model. And when zooming in the upper part, this is how we generated the individual building blocks of Enzovibep binding to the spike protein. Starting with ribosome display, there we selected on different domains of the spike protein. So we had the RBD domain, we had the S1 domain, and we had also the full trimeric spike we selected against and pulled out then a few thousand individual binders. These could be binned with HDRF into different binding categories, binding to different domains, and also we checked with HDRF if they neutralize DNA interaction with ACE2. We characterized all the binders found here, the monovalent building blocks in detail, and also checked for pseudovirus neutralization. And then with the best ones, we went into the bottom part here, where you see the assembly process of the multi-domain DARPin, where we basically took the best neutralizers from the mono building blocks and assembled them first randomly into a trivalent format, where we could basically map out which of the combinations worked best and neutralized best. This was also done in a pseudo-type neutralization assay. And then the best combinations and the best binders went into panel F, which is then the rational design where 22 multi-domain DARPins were assembled. There they had the HSA binders at the N term, and then 3 of the spike binders at the C term. Again, these molecules were then characterized in detail, so biophysics and also the virus neutralization. And the best 2 candidates were the MP420, so Enzovibep, which was moved forward into a hamster model, and hamster proof of concept shown in panel H. With this, I'll move on to the therapeutic design of Enzovibep and why it brings highest viral neutralization. Here we see cryo-EM data, so the individual binder of Enzovibep binding to the RBD open conformation, which is shown in panel A. The DARPin is shown on top. That's the pink molecule binding to the RBM loop of the RBD domain. And below the trimeric spike, you see the top view, and you see that the individual DARPins do not clash with each other, so they combine the open conformation of the RBD very nicely and simultaneously on the trimeric spike protein. In panel B, then the overlay with the ACE2 interacting to the RBD, and here we see this clash of the ACE2 interaction, DARPin and RBD, and this then also proves the neutralization power of the DARPin when bound to the RBD. In panel C, we modeled the linker into the picture to basically measure if the linker length of between the DARPins is long enough that this allows binding of the trimeric spikes simultaneously. If this in nature really happens like this, or whether one of the DARPins also sometimes grabs one of the neighboring spike proteins, we don't know, but we wanted to ensure that the molecule is able to bind with 3 DARPins on the spike protein simultaneously. This cooperative binding was then analyzed in SPR. On top, you see the binding traces on SPR of the 3 individual RBD binders incorporated in Enzovibep. This was on an immobilized, on a chip with the RBD domain immobilized, and you see some off rate when then linking these 3 domains in Enzovibep. you see that the trace, when measured over 10 hours, the off rate on an SPR, you see that the trace does not come down. So basically showing a very high affinity and cooperative binding of enzovibep against the RBD domain in an SPR chip, and this very high binding and neutralization of ACE2 then also translates to very high potencies in vitro. So on the left side, you see the titration curve and IC50 values of 420 and 423. So both of our candidates, and they neutralize in the range of 30 picomolar, which translates to about 3 nanogram per milliliter. So in a very high potency range. Also in vivo, we looked at survival curves in the Roborovsky dwarf hamster model compared to the Syrian gold hamster that many groups use. The Roborovsky dwarf hamster is highly susceptible to the virus, to SARS-CoV-2, and usually when not treated, that's what you see with the PBS placebo-treated curve, hamsters need to be taken out of the study between day 2 and day 4 because they get very, very sick. None of the treated animals with 420 or 423 had to be taken out when administered between 0 hours and 24 hours post-infection. So this was a nice confirmation that we can protect the animals from the SARS-CoV-2 infection. Also in terms of viral loads in the lungs from hamsters taken out at day 3 and day 5, on the left-hand side, we see the black dots for enzovibep, where we see a reduced genome copy numbers measured by qPCR compared to the placebo in red. And on the right-hand side, we see that no infectious virus could be detected at day 3 in the lungs or at day 5 in the lungs, while there were quite high titers detected in the placebo group. With this, I briefly want to come to the protection against variants of concern and also protection from viral escape. There we also had 2 different sites that analyzed our molecules on different variants. So in the table here you see different variants analyzed. So UK, South African, Brazilian, California, New York, Indian variants. So basically everything, all variants of concern at the moment. And in blue, you see the IC50 values for enzovibep, where for all of these variants, we obtained IC50 values in the single-digit nanogram per milliliter range. And this was definitely one part was the design we have chosen with this tri-specific binding to the SARS-CoV-2, as we see on the next slide, where we analyzed the different domains of enzovibep. So we had point mutations, which is in the first column, pseudotype variants with point mutations in the region of the epitope of the individual binders of enzovibep. And then blue is the IC50 value of enzovibep. And we see that enzovibep has only one liability, that's in the phenylalanine at position 486 when changed to a valine, or also to other residues like leucine. And then the white columns are the different RBD binders, so RBD1, 2, and RBD3. And there we see that despite having liabilities on the individual RBD binders as marked in yellow, we do not see a drastic decrease in potency for the full tri-specific molecule, which speaks definitely for the design chosen for this molecule. However, the liability on the phenylalanine 486 remains. Luckily, this mutation is not occurring very frequently. in nature, because this is also one of the key residues that the virus uses to bind to the human ACE2 receptor. And therefore the virus does not like to mutate there unless it loses in infectivity. And we see this on the next slide, where we mapped out the cases on the upper right side, the cases of mutations in position 486. Which is not very frequent. These are the cases in the entire GISAID database. So the frequency is continuously going down over time, and today, so we continued with this trace. We have about a frequency of about 0.01%, so 1 in 10,000 sequences having this mutation. So it seems to be quite conserved, as the virus needs this residue to bind to ACE2. We then also analyzed in a viral passaging experiment with authentic SARS-CoV-2 how long does it take and how many passages to passage on the virus under therapeutic pressure. And for that, we used some monoclonal antibodies, so the 2 Regeneron antibodies and the VRS-309, and compared this to the monodomain DARPin, as well as the multi-domain DARPins, and the combination of the multi-domain DARPins, as well as the combination of the 2 Regeneron antibodies, and we obtained a similar finding what Regeneron has also published last summer. So you see the first 2 passages on top and the passage 3 and 4 on the bottom, and basically I don't want to go into detail here, but what you see is that the monoclonal antibodies lose potency in the early passaging round, so passaging 1 to 3. While the multi-domain DARPins, when looking at passage 4 at the lower left corner, still remain potent, so the virus does not acquire any escape mutations for MP420, 423, or the combo thereof, as well as the cocktail from Regeneron, which also remains potent in passage 4. This was also nice confirmation that our multi-specific approach behaves like a cocktail of 2 well-chosen antibodies. With this, I come to the summary of the achievements for Ensovibep, but do not go into that for time reasons, rather go on for the outlook of Ensovibep and generally DARPin antivirals. Here you see the draft development plan for Ensovibep. We have initiated a phase 1 in healthy volunteers at the end of last year. There we could see good safety profile, as well as exposure of 2 to 3 weeks in human. Then earlier this year, we initiated a phase 2A in ambulatory patients. And in May, together with Novartis is conducting this study, empathy trial was started in ambulatory patients with mild to moderate symptomatic COVID-19. That's a phase 2/3 pivotal study where in the part A, we look at 3 different doses, 75, 225, and 600 mg. Flat dose, and then there will be an interim analysis, which hopefully might lead to a possible EUA status or filing towards the end of the year. And then the best dose will move forward into part B here. Then together with NIH, here NIH is conducting the ACTIV-3 trial in hospitalized patients. There will be a part A with 300 patients at 600 milligram flat dose, and a futility analysis after part B to then after part A, and then to move into part B with 700 patients. With that, also tech development is continuously ongoing. To wrap up, we think DARPins have great opportunities in virology, as seen from this our first virology program, and we would like to dig deeper into various buckets: respiratory viruses, pandemic readiness, viral reservoirs, and also being able to provide therapeutics for lower income countries due to low manufacturing costs. With this, I come to the end, and I would like to thank everybody who collaborated. This is in a chronological order. So Spiez Laboratory and the University of Utrecht, they helped greatly in the beginning, and I've also seen John Bilello and Peiyong Shi in the audience. They also helped in the beginning of this project to move it forward. Then also the Swiss Federal Office of Public Health, they reserved some doses early on, which was great support to move into development. Free University of Berlin, they're running the Roborowski dwarf hamster studies. Then our partner Novartis, which greatly supports the development of Ensovibep and potentially then also does the marketing launch. And NIH, the active team with active-3 clinical trials, but also preclinical assessment at Carol Weiss Lab. And of course, also the entire COVID team of Molecular Partners. I think at least half of the people at Molecular Partners were somehow involved into development of Ensovibep so far, and pretty exciting for us. With that, I'm at the end. Thanks for your interest in the talk. Happy to take questions.
Thank you very much for the exciting results, Marcel. There's a couple of questions around immunogenicity. First, can you say a little bit more about the potential risk that anti-drug antibodies may develop, especially if you have to dose more than once?
Yeah, I don't see you anymore. Yes, immunogenicity is often a question that we're asked because we're not an antibody company and have another scaffold protein. We have seen, for example, in MP250, that's a 4-domain DARPin, where we have analyzed, I think, up to 80 patients, and only a few of them had ADAs, none of them were neutralizing, but that's also not playing with the immune system. For Ensovibep, it's still too early to say something there. What we have seen from the phase 1 trial in healthy volunteers, we have seen very good exposure in humans for 2 to 3 weeks. So exposure is there for treating patients, but it's too early, and the phase 1 is not wrapped up yet to tell more there.
Okay. The other question is around can you clarify how you actually use reactivity studies to look for?
I apologize, I could not hear you well. Could you repeat again?
There was a question about the potential role of immune responses in post-vaccine pericarditis. Clarify.
Apologize, I can't hear you. Maybe somebody else could read it, or. Peter?
Can you guys hear me?
Now it's better. Now it's okay, yeah?
Yeah, the question was about the risk of myocarditis and pericarditis post-vaccine and how you guys screen for tissue cross-reactivity with DARPins.
Yeah, tissue cross-reactivity we used, I mean, we did several tox studies, but also a retrogenics chip. Maybe you're familiar with this technology. That's retrogenics offers a chip-based platform where you can assess binding to any of the extracellular and some soluble human proteins, and there we did not obtain any binding hit on this panel. And then we have also the tissue cross-reactivity panels we looked at, so this all looked very clean.
Okay. So I'd like to thank again Marcel and Lynn for very interesting talks. Thank you very much.